Welcome to Ajat Bioacademy. Today I will talk about intracellular receptor. You can include here cytoplasmic receptor or nuclear receptor, or combinedly you can say nuclear receptor. How? You will get the answer from my lecture. Okay, so the title is intracellular receptor. Okay, and under this is cytoplasmic receptor, and another one is nuclear receptor. Okay, that is, or you can say, nuclear receptor. For a particular region, cytoplasmic receptor can also be considered as a nuclear receptor. Okay, so both are under the intracellular receptor, as the receptor is present within the cell. So intracellular receptor. Okay, why it is cytoplasmic receptor can be considered as a nuclear receptor, I will explain. So I will try to explain with a molecular mechanism of drug that will just work based on the intracellular receptor. Okay, so stay with me. So firstly, I will draw a cell like this. Is, you can consider as a cell, and this is your cell membrane. Okay, and here one receptor is present within the cell, and you can consider this is a ligand. Okay, and that ligand is glucocorticosteroid. Okay, so simply I just noted it by the G, and this is glucocorticosteroid receptor, that is GR. Okay, so because of its basic character, hydrophobic and also molecular size, it can easily cross the membrane. Okay, so multiple number of ligand will enter into the cell, and it will bind with the receptor. Okay, so here you can also you can draw another receptor, and it can bind with the ligand, that is glucocorticosteroid, and here is another receptor. So if you think, before binding the ligand, the receptor is attached with the special type of protein that is called HSP90. Also, it may have HSP70, but mostly HSP90 is mostly functioning here. So without binding, that means before binding, the glucocorticosteroid hormone receptor is present within the cytosol, that is cytoplasm. So it's now in free state. So when the glucocorticosteroid will bind here, then it will change the conformation. Conformational change will happen. Then what will happen? Here, the HSP90 will detach. So HSP90, hits of protein 90, is another name you can commonly you can say this is the chaperone protein. And you know the function of chaperone protein. It can modify, modulate the structure, or it can make the stability of some proteins. Okay, so what will happen when it will bind? Then because of the conformational change, the HSP90 will detach from here. Okay, so then what will happen? The detachment already done, so you can draw like that. And here one glucocorticoid is already bound. Okay, so then what will happen? Instantly, another special type of protein will come here and will bind. That is called P23. And the P23, you can call it co-chaperone. and it has multiple function, because the nuclear receptor, glucocorticosteroid receptor has multiple domain. Already I said must have two domain, that is binding domain and the response domain. But here you can I just roughly draw. This is a nuclear receptor, and here your N-terminal domain. N-terminal domain, you can say this is the DNA binding domain. You can say this is the hinge region, that is linkage region, hinge, and you can say this is the ligand binding domain, and you can say this is the C-terminal domain. Okay, so this is a nuclear receptor, and it is folded in that way, and so that domains are there. Okay, so binding domain already, ligand binding domain already, ligand is bind, ligand already attached with the pocket, and already now what will happen, the P23, it will just express a particular sequence in the N-terminal region. That is, here I can draw here, this is NLS. Okay, that is nuclear localization sequence that is expressed within the receptor, that is the domain of the N-terminal domain, by the help of P23. That means when ligand will bind to the receptor, HSP, that is chaperone protein, will detach, and instantly the co-chaperone protein will attach with the ligand binded glucocorticoid receptor. Some journal says already P23 can also be present at a time, but when it will detach, it will be functioning. But mostly it will detach and then it will attach. This is the most common. Okay, so when P23 will attach with the receptor, then it will help to express the NLS sequence, and also it will make a dimer. That means that ligand binded glucocorticoid receptor will come here, and it will make a dimer. Okay, it will make a dimer. That is a dimer. Okay, so the dimer P23 will help to form a dimer of the glucocorticoid receptor, that is GR receptor, and also it will express the NLS sequence. And then what will happen after that, its main target is to reach to the nucleus. Okay, the main target is reach to the nucleus. So how it will translocate from here to there? So another protein is very important. That protein you can consider, like it will just attach with the NLS sequence of that receptor. That protein you can consider as a importin, and that is the member of importin beta. So some importin protein will attach with the NLS sequence. That importin protein you can say EPO5, you can say EPO55, that will attach here, and it will make the translocate from here to towards the nucleus. Okay, and here the DNA binding domain also there. So here within it will just move towards the nucleus. Then what will happen? The nuclear membrane has a particular nuclear pore complex, and through this pore, the dimer along with the importin protein will enter, and P23 will detach. Okay, sometimes it can enter, but mostly it will detach. So here the genetic materials DNA is there within the nucleus, and the dimer is already just enter into the nucleus. So that dimer carrying the ligand. So here one, and here another one carrying the dimer. Okay, and had some important there, and it will just manage to attach with the genetic material where the DNA is very long. So where it will bind? So some sequence, specific sequence within the DNA, which is mostly selected for the glucocorticoid receptor domain dimer. Okay, so that region, that sequence, you can consider this sequence is for the DNA binding sequence is DNA binding domain is present within the receptor, and that sequence is mostly for the binding. So that sequence you can consider as a glucocorticoid response element. Okay, so over there it will attach. Okay, and that receptor, dimer receptor carrying the glucocorticosteroid, which will just attach with that region, like you can think here. Okay, and it will be functioning. How it will be functioning? It will work as like a transcriptional factor that will express the help to express the gene, or that will suppress the gene, that will just inhibit the gene. Okay, so as this is a glucocorticosteroid, you know it has a strong anti-inflammatory activity, so it will just inhibit the inflammatory gene. It will inhibit the inflammatory gene, negative impact inflammatory gene, and it will just activate anti-inflammatory gene, and that will give its function. After that, it will be translocated by different mechanism. Okay, so this is all about the cytoplasmic receptor, you can say, and that receptor is main target to give its function on the nucleus, so you can consider as a nuclear receptor. And ultimately, you know, this is the intracellular receptor. Okay, so this is all about that matter. So just I can shortly explain. After entering the ligand, it will bind with the receptor, and that receptor already bound with the HSP90, and which is very much important for keeping it within the cytosol and making it stable. After binding with the ligand, the native conformation will change, and then HSP chaperone protein will detach, and then instantly P23 co-chaperone protein will attach, and it will manage the whole stability, conformation, dimer formation, and special types of sequence exposure. And then it will just manage the important protein that will bind with the NS sequence, and then finally it will translocate from cytoplasm to the nucleus through the nuclear pore complex, and then the dimer will attach with the special sequence of the DNA that is called GRE, that is glucocorticosteroid response element. Okay, and then it will give its function. Okay, this is the intracellular receptor or cytoplasmic receptor, or in a sense you can consider nuclear receptor. Okay, so now I will come to the true nuclear receptor. That means receptor already present within the nucleus. Now I will shortly explain. So this is the cell, and you can consider this is nucleus in bold way, because this is intracellular receptor. So this is the genetic materials over here. Okay, and a receptor is present within the nucleus, that is here within the nucleus. So a nucleosome the nucleus receptor is present, and already you can consider as a triiodothyronine receptor or the TR receptor. That means TR receptor. So that is T3 receptor. T3. You know the active form of T4, that is T3. T3. That T3, the triiodothyronine, will just easily pass through the membrane and then finally reach to the nucleus, and it will bind with the. So you can just give the structure is in black color. So this is the T3, and T3 bind with the TR receptor. Okay, so it has, this receptor has also different types of domain, and that will bind with the corresponding sequence. Already, as I said, the DNA binding site was present on the nuclear receptor. So this nuclear receptor that is TR receptor, after binding the ligand, it will make a dimer, as like the glucocorticosteroid receptor within the cytoplasm. So it will make a dimer. Okay, it will make a dimer. So dimer formation done. Different types of factors are there. So dimer formation done. So then what will happen? The dimer. So what is the receptor? That receptor actually not the TR receptor. This receptor is basically RXR. That means retinoid X receptor. That means T3 receptor ligand binding, already attached with the TR receptor, and it will form a dimer with the RSX receptor, retinoid X receptor, and that will translocate from the nucleosome to the particular sequence of the DNA, and that sequence name will be changed according to the triiodothyronine. That means that was GRE, now it will be TRE. That means triiodothyronine response element, and it will attach. So you can attach here this receptor dimer like that. Okay, and it will give its function. So this is the nuclear receptor, because the receptor already present in the nucleus. That is the nuclear receptor. You can consider also the vitamin D receptor also there, that will work on the nuclear receptor. So the receptor is present within the nucleus, so easily you can send the nuclear receptor, and just previous one, that is GR receptor, that was present on the cytoplasm, and ultimate goal of the nucleus. So that was also considered as a nuclear receptor. Both are present within the cell, both are intracellular receptor. Okay, so the cytoplasmic receptor, another example you can consider the cortisol, estrogen, progesterone, like that. That is corticosteroidal hormones or steroidal hormones are working on the cytoplasmic receptor. And here the T3 receptor and also vitamin D3 receptor.